Hello everyone, and welcome to today's live broadcast, Applications of Targeted Gene Panels in Inherited Disease Research. I'm Julianne Chayet of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by the Clinical Sequencing Division at Thermo Fisher Scientific. For more information on Ion's Warrant Next Generation Sequencing products, please visit thermofisher.com/ionswarrant. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button on the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button on the lower left. We have a poll that we'd like you to answer, and the question is, what disease area is your current research or testing focused on? The poll has been closed. The webinar, 49.5% of you have said inherited diseases, 34% have said oncology, 8.7% have said infectious diseases, and 7.8% says reproductive genomics. Now, this webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. I would now like to introduce today's speakers, Brian Meyer and Robert Sebra. Dr. Brian Meyer is the chairman of the Department of Genetics at the Research Center of King Faisal Specialist Hospital and Research Center. For the past 2 decades, he has primarily studied the genetic basis of recessive Mendelian diseases. He has authored more than 100 related articles in peer-reviewed journals. Scientists in the Department of Genetics have used his findings to develop molecular genetic testing specific to Arab populations. Dr. Meyer is one of the founding scientists of the Saudi Diagnostic Laboratories and the Saudi Human Genome Program. Through the Saudi Diagnostic Libraries, he has introduced the region to cutting-edge technologies in molecular diagnosis, carrier screening, and preventative procedures. The Saudi Human Genome Project is targeting the sequencing of over 100,000 samples over a period of 5 years. The objectives of this program include capacity building in genomics, characterization of rare diseases, profiling of cancers, and development of pharmacogenetics to set the foundation for personalized medicine. Dr. Meyer's current interests include the study of genetic basis of more common polygenic traits such as type 2 diabetes, cardiovascular disease, and autism. Dr. Robert Sebra is the Director of Technology Development at the Eken Institute of Genomics and Multiscale Biology and an Assistant Professor in the Department of Genetics and Genomic Sciences at the Eken School of Medicine at Mount Sinai in New York. His work has led to over 30 publications and 4 patents. Dr. Sebra has vast experience in various molecular, biochemical, and sequencing technologies. More specifically, his expertise includes photopolymerization, chemistry, surface bioconjunction techniques, lab-on-chip platforms, active biomaterials, single-molecule detection assay development, and integration of DNA sequencing technologies for scientific and clinically driven applications. Since establishing a technology development team at Eken School of Medicine at Mount Sinai, Dr. Sebra has applied an integrated genomics approach for better infectious disease surveillance. The team works to understand genomic identity between community and hospital isolates by comprehensive characterization of microbial isolates. They have additionally initiated research to derive better targeted approaches for human disease diagnostics that relate to cancer and inherited diseases using both long and short-term approaches. Short-read approaches. He currently pioneers research integrating both short and long-read sequencing techniques to address human disease, both inherited and somatic. He is working toward the integration of bulk and single-cell methods to create niche clinical diagnostic pipelines. I will now turn it over to Dr. Meyer and Dr. Sebra for their presentation.
Thank you, Julianne, for that kind introduction, and a good day to you all in whichever time zone you are joining from. Today I would like to talk to you about the development of targeted gene panels for Mendelian disorders research as part of the Saudi Human Genome Program. What you should now see is the characteristics of the Saudi population. The Saudi population is largely tribal in its origin. A number of tribes enumerate more than 250 individuals per tribe. However, for the purposes of this presentation, the two features that are most relevant are consanguinity and large family size. The Saudi population, the consanguinity rate is about 57%. However, when you go to many of the specialty clinics where you encounter inherited diseases, up to 98% of patients come from parents with a consanguineous background. The large family size is also extremely useful when it comes to mapping inherited disorders. As one would expect in a population with such a high consanguinity rate, there are many novel autosomal recessive disorders, and in being able to access families where there are 3 or more affected individuals, it provides significant power for mapping these disorders. So here you now see a consanguinity map of the world with Saudi Arabia in the center. It's quite clear that the Saudi population has one of the highest consanguinity rates in the world, and associated with that, of course, is a very high incidence of rare recessive diseases. You now see a list of the monogenic diseases traits as they were discovered over the years, starting in the 90s and going through to 2012, with 2012 actually showing there being some, if I can get my arrow working here, some nearly 4,000 to 5,000 monogenic diseases, many of them autosomal recessive. So the Saudi Human Genome Program is a program that was initiated some 18 months ago, and is a national program which aims to sequence 100,000 samples, both normal and pathological, from the Saudi population over a period of 5 years. Some of the relevant objectives of the Saudi Human Genome Program were to sequence thousands of gene panels, exomes and genomes over this period of time in order to build capacity in genome scale sequencing and to catalog genetic variants in the population, both normal and those associated with a number of traits. And in doing so, to become a global leader in genomic medicine, particularly in relation to inherited disorders, which are particularly frequent within the Saudi population. In 2013, it was understood that there were some 4,800 diseases with a known genomic cause, and an approximately equal number where the cause was unknown, with up to 2,000 diseases where the underlying gene was still to be discovered. With this background, we set out to study inherited diseases as part of the Saudi Human Genome Program, and clearly with the tens of thousands of individuals that we had access to, it became apparent that to simply run exomes in all of those individuals may not have been such an efficient strategy, particularly when essentially one is looking for a single variant causing an autosomal recessive disorder. At that time, we took a decision to develop multiplex panels of genes to cover all known Mendelian disorders, and we coined the frame Mendeliome. The idea here was to cluster genes into panels in a meaningful manner based upon pathology and function. So, for instance, we developed a neuro panel, a renal panel, pulmonary, immune deficiency panel, etc. In each of these panels, we wanted to cover the coding region and flanking sequence of each of the genes in that particular panel. So a Mendelium gene panel was based on the decisions of two medical geneticists who looked at all of the genes in Omum and then clustered them into 13 panels. At this time, which I think was in late 2013, there were some 4,300 monogenic disorders and some 3,200 genes in Omum underlying these disorders. So the 13 panels encompassing all of the Omum genes were defined broadly based upon target organs with some redundancy in gene content of individual panels. For instance, some genes would appear both in the neurological panel and also in a dysmorphia dysplasia panel. We developed multiplex gene panels using Ion AmpliSeq designer software. The primer design was based upon generating amplicons that had an average length of 200 base pairs. This was given that we expected to get a good quality of sequence with that read length. With the current technologies available to us in chemistries for the ion torrent system, this read length, of course, can now extend up to 400 base pairs. We also wanted to have a minimum of 90% coverage of the CDS, and for that to include 10 base pairs flanking each of the exons. Essentially, we took the decision to only focus on the CDS and not to include the 5' and 3' UTRs, because by adding 5' and 3' UTRs, this significantly increased the number of amplicons required in each of the panels. And so there were both some cost factors involved, but also we felt it would be more efficient to focus completely on the CDS. Following the in silico design, the coverage was assessed for compliance with design criteria and manual processes. Essentially, the bioinformaticians looked at the predicted amplicons and checked for coverage of the CDS and flanking regions. They also, at that time, checked for, instance, any interaction between primers and 3' SNPs that could impact primer efficiency. Following the in silico design and evaluation, primers for each panel were synthesized and pooled into two multiplex reactions. These two multiplex reactions were based upon PCR compatibility and minimizing the likelihood of primer-primer interactions. Following synthesis, primer pools were tested for coverage and recommended multiplexing and other QC metrics to ensure specification were met. Essentially at this time, for the larger panels, some of these panels, as you can see from the next bullet, included 758 genes, while smaller panels were down to 96 genes. And in the initial testing of panels, the Ion Torrent team gave us some indication of the number of samples we could multiplex depending on which chip was being used, either on a PGM platform or on the Proton platform. So generally, the panels, we achieved a coverage of 97 to 100% of genes in each panel, greater than 90% coverage for 97% or 100% of the genes in each panel. Here in some sort of largely small figures, you can see some of the characteristics of our amplicon design. You will, for instance, see the amplicon length, which we targeted 200. So you look down that column, you will see that we generally clustered around that 200 area, particularly when you look at the average amplicon length. The average amplicons per gene were around about 20, and the number of genes ranged from 758 down to 96. You can also get some idea of the number of bases covered for each panel. Essentially, these parameters helped us decide the level of multiplexing that we could use when testing our samples with these panels. In general, for the larger panels, such as the neuro panel and the dysmorphia dysplasia panel, we tend to multiplex up to 24 samples per run. With smaller panels, such as the renal panel, we have multiplexed 48 samples, but could easily do so with up to 96 samples per run. Here are the typical results that we achieved with our panels. In this case, the neuro panel. The number of genes in this panel, it says 757. The average gene coverage was 99%, meaning that 99% of the CDS and the flanking regions were covered. The median gene coverage is 1. The fraction of genes covered at greater than 90 and 95% were 0.99 and 0.97 respectively. So essentially, I think the expectations from our design were exceeded substantially. As you may recall, we targeted 90% coverage of the CDS and flanking regions, and in fact, for greater than 95% of our genes, we were achieving a figure of 97%. Typically, for our gene panels, in this case looking at the dysmorphia dysplasia panel, a panel that contains 422 genes, we were getting coverage in the order of 400x, as you can see from the average base coverage, which I have just marked with an arrow. The workflow for running our gene panels, on the left you can see the basic analysis workflow, which was the base calling and quality check, which resulted in an unaligned BAM file. Then mapping to the reference genome to create the BAM file, which you can see here, followed by variant calling for the initial VCF file, annotation of that VCF file, and then filtration of that VCF file to get down to variants of significance. With respect to our filtration, on the right-hand side, you can see the process that we used. Firstly, the filters removed deep intronic, synonymous, UTR SNPs, and non-frame shift indels. We also removed frequent variants in public databases, removed variants that we found within the Saudi population, then removed low confidence variants based on a quality score from the Ion Torrent software, and this usually came down to providing us with well under 20 variants, and often 1 to 2 variants from which we were able to look for variants associated with a particular trait that we were investigating. Here now you get some idea of the results that we had across all of our 13 panels. On the left, you can see the approximate number of variants that we got per sample in each run. When you filtered this out to remove any non-functional variants, this figure was decreased in each instance. Further filtration to remove variants that are polymorphic, as represented in the population-specific databases, reduce the number of variants further. Then filtering against variants specific to the Saudi population. You can see now we are getting down into well under 50 variants, even for the larger panels. A filter used to remove variants that had a quality score less than 100, now brought it down in some cases to single digits. And in the Saudi population, where one would expect that the causative variants are homoallelic, we could use zygosity as a final filter. And by using zygosity as a final filter, you can sort of see in many cases, for a lot of the panels that we were running, we often got down to a single variant. Typically here, you see represented one case from our neuro panel. This is a run in which there were 24 panels covering 758 genes, or just over 18,000 amplicons. We had an average of 162x coverage that we received for this particular run. Typically, we were getting around about 1,106 variants in our annotated variant file. Removing out poor quality variants, this was reduced down to 717, as you can see here. When you take out when you take out variants that are non-coding or not splicing variants, down to 643, removing any synonymous variants brought it down to 263, taking out variants from public databases down to 21, and finally using zygosity was able to reduce down to a single variant. So essentially, from the last few slides, our expectations of being able to use panels to, one, not be overwhelmed by the number of variants that we started with, and to be able to interpret the variants using targeted gene panels, we were almost always able to get down to a handful of variants that we would then use an expert opinion to associate with a particular trait. So applying this Mendeliome assay, as we coined, we wanted to look at the analytical sensitivity and the clinical sensitivity. And when we talk in terms of the Mendeliome assay, it was our view to apply the 13 gene panels where, based on the primary symptom or primary trait associated with a particular sample, one would automatically assign that to a panel. For instance, if the trait was intellectual disability, it would automatically be associated with the neurological panel. Similarly, if an individual had a movement disorder, the genes associated with movement disorders were also in our neurological panel. Initially, we looked at 642 samples with known mutations that had previously been identified by Sanger sequencing, and our panels were able to pick 79% of these mutations. 46%, or 62 of the negative cases, were due to a design flaw. And when I say design flaw, I simply mean that the sample was not assigned to a panel which contained the gene which had the causative variant. So this is a situation where the redundancy in our panels was not large enough, given the strategy that we used to assign a sample to a panel based on a primary symptom. If you removed this design flaw, 87% of the variants identified by Sanger sequencing were immediately picked up by the ion torrent panel. Essentially, single nucleotide variants, 93% of them were identified. For indels, the rate was somewhat lower at 72%. When we compared the ion torrent panels with data that we had from our SNP genotyping using an Axiom array, we found 95% concordance with the Affymetrix calls, and there were 30 additional SNPs that were called through our sequencing but failed on the Affymetrix array. So to summarize, the Mendeliome assay analytical specificity for SNVs was 93% in our initial run. This was an initial run of 2,500 samples. We have now gone to nearly 10,000 samples and are using the newer chemistries, and we believe the rate for SNVs is actually much better than that. In our initial run, also the indels, 78% of those were picked up through ion torrent sequencing. To look at the Mendelium assay clinical sensitivity, on the left side you can see the various panels: cardiovascular panel, deafness, etc., etc. You can see the overall sensitivity, which is in this column. But the important message here was that within each of those panels, the sensitivity of specific subgroups, for instance, if you looked at movement disorders in the neurological panel, at 69% can be higher than the overall average. I would like to draw your attention to the genome biology paper in which all of this data is published. I would invite you to review that at your leisure. Here again, results for the last 4 panels, again, essentially with the same characteristics, where the average is presented in the column in the middle, but you can see the sensitivity for subcategories within each of those panels. Specifically, Sanger confirmation of a likely variant, based on Sanger confirmation of a likely variant, the overall clinical sensitivity of the gene panels was 43%. When you compare the Mendelium assay to whole exome sequencing, there were 213 samples that were negative in the Mendelium assay. We decided to run them with whole exomes to actually see if whole exomes would provide the answer, or to see what proportion of samples whole exomes would provide the answer. At the time, there were 170 additional genes in OMIM that were not included at the time that our panels were designed, and this could be one of the reasons why the Mendelium assay was negative. We first ran the 213 samples on an Affymetrix cytoscan array, and in 35 of the samples we found the cause to be a de novo pathogenic CNV. We ran whole exome sequencing on the remaining 178 samples, and pathogenic variants were identified in 20 or 11% of these 178 samples. Interestingly, 70% of those 20 samples, once again, the variant was missed because of a design flaw, namely the gene was not present in the panel on which the sample was run. In 6 cases, the gene was not present in the Mendelium panel in total. So in essence, whole exome sequencing only really rescued for us 6 cases out of 213. And interestingly, 2 cases were missed by whole exome sequencing, but were detected by the Mendelium assay, and essentially was because of interpretation and a lower coverage in running the exome sample. The Mendelium assay did for us give us some additional insight into the clinical spectrum. Here, for instance, with BBS4, which is associated with Bardet-Biedl syndrome, we were found a mutation in BBS4 which was associated with isolated retinitis pigmentosa. So the Mendelium assay, there were many examples where it helped us expand the clinical spectrum of the research samples that we were processing. The Mendelium assay also improved annotation of the human variome. We were able to confirm many of the candidates in OMIM where there was a single allele present. Examples you can see in the first bullet. There were 433 novel mutations in a total of 788 variants associated with Mendelian disorders, and this was the largest number from a single study, and it was based on the 2,500 samples we initially ran. We found a very large number of polymorphic variants attributed to individuals of Arab ethnicity, and it showed us how poorly represented this ethnic population was in the genome databases. Our study identified also 342 HGMD variants, which were thought to be disease-causing, but clearly in this population were polymorphic, and this requires some adjustment in HGMD. Similarly, in some instances where minor allele frequencies in the 1,000 genome database were less than 1%, these were significantly greater than 1% in the Arab population. In summary, the Mendeliome assay gave us a yield of 43% versus a whole exome sequencing that averages around about 25%. Some of the higher yields were present in our pediatric ophthalmology panel and for neurological disorders. The specificity of presentation impacts on assay sensitivity, I think we covered that previously, naming some subcategories of disease had a much higher clinical sensitivity than others. Our WS studies often identify mutations in known genes. This was seen through 213 samples that were negative on the Mendeliome, but then found to be positive in the exome for genes that were present within our Mendeliome panels. I would like to acknowledge the team who are listed here, and you can review at your leisure. And finally, acknowledge the Saudi Human Genome Program. Thank you for your attention.
Great. So thanks for the fantastic presentation, Brian. This is Bobby Sebra speaking from Mount Sinai. Now I'm going to switch to my content and kind of piggyback off some of the information from Dr. Meyer's presentation. So today I'm focusing on the high-throughput comprehensive screening, essentially also using AmpliSeq technology as well as the S5XL system in the Torrent platform. And I will mention both Mendelian and hopefully have a little bit of time to discuss how we're utilizing the OncoMind panel as well, since it seemed like about a third of you were interested in some of the cancer genomics. Most of the work that we'll be presenting today is research use only, but it is implied that we would be pushing this forward into clinical use. So to give you a quick overview of what our institute utilizes in terms of a toolbox for addressing a vast array of disease within human health care here at Mount Sinai. To give you a scale, we tend to have millions of patient visits in our hospital system per year, so you can imagine the diversity of disease that comes through our system, therefore the need for a variety of technologies, where as many of you are probably accustomed to using Illumina platforms for whole exome, whole genome, and RNA-seq, we do that as well. But we focus primarily using the ion torrent systems to basically then focus on targeted panels, much like Brian presented. And as you can see, we have a nice arsenal of these technologies spread from 10 ion protons to now adopting 5 S5XLs, 2 PGMs, and 11 chefs to enable us to do this at high throughput. And I'll discuss how we're applying that. On top of the short-read technologies, we do tend to use a lot of long-read sequencing as well, because as you know, there's no such thing as a one-size-fits-all sequencing technology right now, so this allows us to plug and play and often validate the results we see orthogonally across various platforms to address both small and large structural variants in inherited infectious and somatic disease. So looking forward, if you look at how we're applying the ion torrent technology across our system, it's pretty vast. So the reason we need all of that equipment is we have targeted panels that are being designed. One I'll speak about today in depth on the next set of slides is essentially the 25,000 amplicon Mendelian panel that we have, which is similar to the Mendeliome panel presented by Dr. Meyer, but unique to our needs within our healthcare system. I'm not going to speak about the infectious disease components listed below, but it is nice to say that we have worked on influenza sequencing using the Torrent platforms, as well as 16S sequencing for metagenomics and RNA-seq. And then in somatic disease, you see a list of research-based, community-derived panels driven by our oncologists here in our healthcare system, inclusive of the OncoMind commercial panel, which is comprehensive, and hopefully I'll have a little time to discuss that later. And we have had success with New York State approval in the commercially available Cancer Hotspot version 2 panel to put that out there, so there is clinical utility in that light. And one other thing I'll mention before going into the research is our switch over to the S5XL instrumentation. I know many of you out there are probably using PGMs and protons right now, so I wanted to give you an overview of how our lab views the benefits of switching over to S5XL, and I'll show the actual data comparisons later on in context to the science that we're conducting. But quickly in this list, you can see a reduction in the initialization time, so that saves the operation time so you can do more cycles per day. You no longer have all those big packages of external disposable supplies and reagents. They just come in 3 plug-and-play reagents that you plug into the device. The chlorite wash, which no one really loves doing to initialize the system, is now removed on a weekly basis such that you no longer have to do that maintenance. And it reduces the hands-on time down to 5 minutes. So the reason that's important to us is when we're operating at high throughput, we really do want to push the S5XLs toward 4 cycles per day instead of just 2. And since this was presented at AGBT, we've actually now done over 50 runs on the S5XL, which is what you'll see some of the statistics presented later from across the panels that you see down at the bottom, which span both inherited and cancer genomics. And you can see the performance in terms of number of reads and quality are very similar between the platforms. So the first scientific use case that I'm going to focus on takes us back to inherited disease, similar to what Brian was presenting in his talk. But specifically in this particular case, I'm going to take you through a use case, which is the Resilience Project, but then how we designed the panel. And I chose this order because the way this panel was designed was working with Eric Schott, Rong Chin, Stephen Friend, and others to basically determine content that would help us interrogate very large populations of individuals on the order of hundreds of thousands of individuals to collect those samples and then look through different layers of Mendelian disorders to look for human resiliency. And by that, what we mean is the ability to identify individuals that have buffering mutations in their genome where in fact these individuals are affected and do have a typically highly penetrant deleterious mutation that would have been associated with early onset before the age of 18 and a phenotypic presentation of the disease by that time in their lifespan, when in fact they're perfectly healthy with no presentation. So by looking for these individuals, it helps us drive the ability to interrogate the information in those genomes and better understand potentially how to get at novel therapies based off of the information gained from discovering these individuals, while also screening the affected population to interrogate and build better databases of genomic information in the loci presented on the targeted panels here. So to do that, Rong Chin spearheaded a really great effort that took note of all of the data that was publicly available down here at the bottom. If you look at my pointer, everything shown in red here basically shows all the databases that he used to basically pull out 500,000 data sets that were microarray and GWAS study data sets, and then almost 100,000 whole exome data sets that were available to do a retrospective analysis. We were taking all of that information and essentially doing a filter with a disease prevalence of 1.25 million with both the penetrance and severity phenotypes that I mentioned previously, to then screen through the individuals to both look where in the human genome we were potentially seeing diseases that were negated by these buffering mutations to help us focus, but also to just look at the statistics and the prevalence across the information. And so what that condenses to is a result that looks something like this, which is a Circos plot that you'll be able to download from his publication when that comes out, hopefully very soon. And so if you look at the different tracks on these individual rings, you can see the breakdown of Mendelian disease, and those of you that are experts know that there are over 6,000 known Mendelian disorders across the human population in various haplotypes right now, but unfortunately there aren't many drugs that cure them. We just have lots of drugs that essentially suppress various phenotypic presentations and symptoms. So in this, if you look at the breakdown of disease and then the prevalence, you don't need to be able to focus on the other rings right now, but what they are is basically the statistics across the human population and the likelihood of being one of those 6,000 diseases or having one of the 150,000 known mutations that are associated with those diseases, but also how they cluster within individual chromosomes to look for, more concentrated loci for targeting on the panel design. But more importantly, something that's interesting for all of the sequencing experts out there is when you really hunker down on this data and you look at the quality across the 600,000 retrospective cases that were utilized, there's tons of ambiguity within sequencing data. So what Wrong did was essentially look at only highly significant cases, which you see in the center of this plot, where we do indeed think that these are very highly likely to be resilient individuals from the retrospective analysis. So it gave us a precedence or an order of magnitude for us to understand that yes, this did exist and we will be able to push. But even though there are only 13 to 15 individuals noted in this study on the retrospective analysis, there were actually more like hundreds to thousands of individuals who were potential resilience candidates, but the quality of the data that was collected on those studies wasn't high enough, so those are left out. You would want to come back and orthogonally validate them or increase the coverage, which brings in the importance of doing some of these targeted approaches that Brian and I are talking about today. So this leads to the actual design of the panel. So at this point, we're now transitioning beyond the retrospective analysis into how did we design this panel. And so the importance of the modularity of the panel that's being presented today is not only did we fill the panel with genes that are of interest to our hospital system, but also to the Resilience Project. And so the stats on this thus far are such that we have greater than 700 genes covered, so it's a very large panel. And when I say that, these are disease-associated genes where we're basically covering, for all intensive purposes, all coding exons. But then on top of that, because there are diseases where there are hotspots with genetic loci that are affiliated with the presentation of disease where you don't need to interrogate the entire gene, both within exons and introns, there are an additional 4,000 hotspots added to this panel. So when you merge the two, that comprises essentially 4.4 megabases of genome, so you can think of it as 5 to 10% of a whole exome, broken down by almost 25,000 amplicons in just two pools. So the two pools combine read lengths from, or inserts from 125 base pairs to 275 base pairs, and the reasoning for that is when we do our 50 base pair flanking on the 5' and 25 base pair flanking on the 3', you're then left with shorter amplicons when you're just doing hotspots, whereas you have larger amplicons when you're trying to do whole exons. And you can look at the statistics in this table above, shown here by the arrow, where basically you can see that our performance metrics, after essentially manually curating the coverage of amplicons on targets, after about 6 revolutions of iterating this particular panel, you could then see that we could get up to about 98% coverage of the desired bases covered in genes, and 86% of the hotspots were covered. So moving forward to the next slide, looking at actual performance metrics of a complex panel like this can be interesting. Not only do we actually care about the individual performance from sequencing and the variability there, but the ability to essentially interrogate all of these amplicons. Because we have 25,000 amplicons there, we wanted to assure that we were operating at a level that allowed us to get good enough coverage to interrogate the variants. And so if you look at the plot shown down here in the bottom right corner, you can see that the on-target is about 97% with 92% uniformity. And so that uniformity is really important because when you look at this purple line, we chose to do 200x coverage along the lines of some of the concepts that Brian talked about because we wanted to have the sensitivity such that we could detect a variety of both indels and SNPs. And so the other thing that's important to note is this plot, if you look at the x-axis, it's just broken down amplicon 1 through 25,000 versus coverage. In most cases, we're actually seeing much higher coverage than that, but we did want to make sure that we were getting a minimum of something on the order of about 50x coverage for the underachieving amplicons that had some bias due to the big complexity of the mixture that's being utilized. So an example of what the target design would look like is shown on this next slide. So if you look specifically at something like P53, which I chose simply because it's important to almost all fields, both for driver mutations in cancer, but also for inherited cancer. This being said, if you look at our coverage of amplicons broken down across the target, when you look at this small red section, the red track at the top, let me see if I can get my arrow working here, that's the only region that is dropping out of the coding region within our design. So this represents kind of what I would consider to be average performance. But for the users out there, if you wanted to go back and actually design primers within that particular region, you could white glove them or manually design primers that would allow you access. So at this point, the design shows that we're seeing about 98% coverage of P53 covered by our panel, but if we wanted to increase that, you can change the size and the location of your primers to do so. And that's the type of thing we were doing on the 25K panel to really push this forward. So what do some of the performance metrics look like, and how are we assessing this? Because in our case, we're looking for variance, but we don't always know ground truth or have a control for comparison. So basically, this table shows, for those of you that have transitioned from the old V3 chemistry to High-Q, if you focus on what's in the purple box here, you can look at performance metrics comparing on our 25,000 amplicon panel what we're seeing in terms of sensitivity, PPV, and the false positive rate, which I think are the most important metrics to look at. Now, our ground truth in this case, we chose to use the NIST genome in a bottle data set of variants. So technically, there's only 3.37 megabases of the 4.37 megabases of the panel overlapping. So essentially 75–80% of our data is then validated, considering ground truth to be the reference. And so that's how these statistics are derived. You can see both the improvement from changing the chemistry to the higher accuracy chemistry, but also that the false positive rate is really starting to equilibrate at a reasonable level at 33 false positives per megabase. And then to give you an idea, it was almost double that on generations of chemistry about a year ago. Now, I will note we did this analysis on whole exome data that we generated as well, and the numbers on whole exome are actually better than the numbers from our panel, and the reason for that is we have intronic regions in our panel, which actually are harder for the torrent technology to achieve in some cases due to the indels and the repeat structure that are in those loci. So I just wanted to note that the numbers are actually about 1% better on the sensitivity and PPV for some of the commercial kits. So I'm going to skip that slide and come back to it in a different light to save a little bit of time. But another important thing to show is that in our case, we're seeing on the order of up toward thousands of mutations, in this case 5,000 exonic missense variants, when you don't filter. So in this particular case, in a given sample run on our 25K Mendelium panel, you're going to run into about an R-squared fit of 93% between the S5XL and the proton. If you start filtering based off of actual sequence criteria and reference statistics to basically prove what's right and what's wrong. The numbers go much higher, so this is basically a worst-case scenario. But even then, you see really nice concordance between the two across the 5,000 missense variants that we saw on the control samples, which in this case, in this case are just Coriell control cell lines that are being utilized. And you see nice clusters of 50%, 100%, and 0%, which you would expect from the alleles in this particular panel. So before I switch gears for about 3 to 5 minutes on cancer, I did want to say that thanks to the Connecticut genomics testing facility that we've built here at Mount Sinai, we're actually really scaling up the efforts. So for those of you curious about how many samples we can produce using a panel like this and how to do so, Todd Arnold and Dan Cisco and others who operate that facility are now operating specifically on the 25K panel across 8 protons, which allow us to sequence somewhere between 192 and 385 samples per day across a fairly small number of FTEs using automation equipment like Hamilton's and so on. So this has ported itself nicely onto high-throughput equipment and allows us to now start generating the data instead of relying on retrospective analyses. We're now generating thousands of samples using both biobank samples and collected samples for projects like the Resilience Project. So a quick overview of some of the work done with OncoMine, both for the reasons of addressing maybe questions that the webinar audience might have, but also to show you some statistics from commercial panels so that you see kind of what you would get if you're using panels that you can buy from Torrent. First, our program at Mount Sinai is incredibly broad in terms of how we tackle cancer. We do everything from develop methods for R&D on single-cell isolation methods and looking at tumor heterogeneity to basic whole genome, whole exome, and then, of course, targeted sequencing, which is how we typically rely heavily on the Torrent platforms. The panel that we have the biggest use case for cancer is the OncoMine panel, which I'm showing here briefly to show you that it has 143 unique genes. We've sequenced hundreds of samples on this panel to date in an R&D setting and have been pretty happy with both coverage and otherwise, but when we have cases that do not have genes covered that we need, we simply make our own targeted panel, which are some of those panels I showed at the beginning. So if you look back at performance for somatic disease using the comprehensive OncoMine panel, you can also see that it's very, very similar to what I previously showed for our Mendelium panel, with one exception, which is the read length plot, which I'm pointing an arrow at right now, is very much a more narrow window. And the reason for that is on our custom panel for the Mendelium, we specifically choose to do hotspots and targeted exons. So we have a mixture of small and large targets on purpose, where in this particular case, on the OncoMine panel, it's pretty much targeted to be mainly exons and a fewer number of hotspots. So that's just designed by the commercial panel itself, but it's much more conserved. But the performance itself, which I think a lot of you should be interested in running a panel like OncoMine, when you're porting from Proton to S5XL, this is what we've seen from the technology in our hands. So you can see that across all variants, on a panel of this size, which is only less than 200 total genes, we're seeing about a 95% concordance between the two technologies, and that's across, roughly 4,000 variants. So we've been pretty happy with these numbers. And then even on indels, our rate is basically a 72% overlap, which may not be perfect, but the number of indels we have is pretty small. So it's not affecting the overall performance. And you can filter many of those out. So if you look at the star at the bottom of the slide, this is an extremely low stringency filter where we're allowing everything through that is 5% or above. And in this particular case, we're generating on the order of 500 to 1500x coverage from sample to sample. And that comes out to be about 1,000 exonic missense variants of which essentially 70 to 75 per case usually have kind of a clinical meaning to them. And then if you go in and you look at the ones with actionability, the variants that are actionable that are discordant between platforms, if you take a closer look, all of them are easily filtered out when you do a manual curation due to things like strand bias, low allele frequency, and some of that is due to the sample itself and the purity. And so when you come back and do this type of analysis, you can actually improve those numbers even more. But it is important to show the raw data to an audience like this because how we all filter our data is really up to our organization. And with that, you end up seeing about 99% R-squared value and a 0.96 R-squared value on the Oncomine annotated variants, which again are the ones that are more likely to be actionable for drug therapies. So with that, I'll summarize by saying, we've hopefully shown you that we can utilize custom AmpliSeq panels and comprehensive AmpliSeq panels for both inherited and somatic disease. And I want to thank a bunch of people, those shown in bold, so Eric Rong, Jason Bobe, Todd Arnold, and Stephen Friend are all very, very strong contributors and leaders for the Resilience Project, which is the biggest use case of the Mendelian panel, Kamada Alicia Hanan, and Wissam for actually operating and running most of the super panel within the R&D setting and the Connecticut genetic testing facility for pushing that through to higher throughput. And with that, I think we'll take questions for both presentations. So I'll hand it over to whoever is curating that. Thank you for your time.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We will try to answer as many questions as we can. There will also be a poll question that we will push and leave open during the Q&A. Our first question is, how do you interpret the sequence variants? Do you have comments for using software such as Sapientia from Kazenta Company to interpret the sequence variants?
Julian, I didn't get that question clearly. Is it possible to repeat it?
Yes. How do you interpret the sequence variants? Do you have comments for using software such as Sapientia from Kazenta Company to interpret the sequence variants?
No, we actually have our own pipeline. We of course subscribe to many different databases during the annotation. The filtration process was in my presentation, and that essentially gets down to a small number of variants where at that point we rely on an expert opinion.
Yeah, and I'll add, from our side, we essentially do almost exactly what Brian has mentioned as well. We also have internal groups, both production and R&D, to annotate those databases, but also use public databases for the annotation. But it is important to filter out the low-quality reads and do that curating first to make your life easier on the annotation side at least. But otherwise, we're doing essentially the same thing as Brian's team.
Great. The next question is, what steps have you taken in case of bad amplicon read quality?
Yeah, so on this end, in terms of bad amplicon read quality, there are a lot of things that can lead to that. That could be if you have dropout in your sample. So if your sample is degraded, you might have regions that drop out. But moving beyond the sample in the sequencing component, if by bad read quality you mean lower coverage, lower QV, strand bias, any of these types of common kind of features, the first thing you can do is when you do your design, the 6 iterations I had mentioned on our panel, we really tried hard to iterate in between in terms of just in silico to make sure the coverage would be there. But when you do a run, if even after computationally you think you have the perfect panel, once you start running it, if you had bad read quality in certain domains, the easiest thing you can do is increase your coverage such that you start to have higher calls in that domain. And if it remains biased, you would actually, in my opinion, go more toward either testing another biological replicate. If that still failed, you would then have to think about potentially spiking in additional primers to try and get better coverage in that domain.
Yeah, I'd agree with that. We've had some instances where we've missed variants that we thought we should have picked up, and on reviewing the amplicon had primers redesigned and spiked the panel. Also, by going for sort of very high depth in some of our panels, we are getting to over 400x, as you saw with our dysmorphia dysplasia panel. I think this is the way to safeguard against having those poor regions for amplification and sequencing.
Great. The next question. Considering the fact that the genomic variation is best seen in non-coding sequencing, why is such data discarded? I believe SNPs or variants from those regions could be important.
Well,
yeah.
I don't think the data is discarded. Of course, our gene panels are largely designed to cover coding regions, but if we do find some variants in the flanking regions, etc., as you do with amplicon design, I think this is just a priority of filtration, and if we remove that, which we consider to be noise initially, it helps us get to the variant that we're looking for. If we don't find that variant, of course we would go back and look at our unfiltered VCF files to see if we can salvage something.
Yeah, and I also agree with that approach. One thing I will add is also, in addition, obviously all of our panels are designed around the coding regions. As you saw, we did insert some intronic variants of known significance, but if you're hunting for variants of unknown significance in introns, it's under-curated in databases, so to do that you just have to have high sample numbers, and often because of the nature or the complexity of the sequence in some of the energetic regions and intronic regions, basically a combination of technologies usually becomes important. So we do some of that on multiple platforms to kind of cross-validate so that we know when something like a de novo mutation is actually useful or not for the cohort of interest.
Great. The next question. What is the possibility to design a panel for trinucleotide expansion disorders?
Yeah, that's a great question. It's actually something that we work on pretty heavily, and you might have seen presented in some of the work we do from Sinai using long-read technologies. So, not to go off topic, but I think it's really important to be able to utilize long-read length to tackle trinucleotide expansion disorders. So it's very difficult to do on short-read sequencing methods. And you also have to work on methods around removing amplification from your system, because often your polymerases die when they're trying to get through GC repeat-rich regions. And so, in summary, I think that's extremely important for those diseases, but also one of the most challenging things to do with standard NGS technologies.
In short, it's something that we haven't addressed with our inherited diseases panel, and it's not something that is high on our priority list at the moment.
Next question. Will all the data analysis, i.e. variant filtration, be supported as a package when using the panel?
I didn't get that, Julianne, once again.
Will all the data analysis, i.e. variant filtration, be supported as a package when using the panel?
Oh, I see. You know, the essentially, you know, we developed our own package for doing the filtration, and you know, whilst it's the annotation that we use through the public domain databases or the subscribed databases is readily available, we also filter against variants that we identified in this specific population. We're very happy to share the information about our filtration, and people can filter against those databases or subscribe to the necessary databases when they conduct their own annotation. With respect to variants from the Arab population, this is something that still is being discussed at the Saudi Human Genome level. It is our intention to make these publicly available for research and academic use.
Next question. How about the analysis complex traits like BMI? What methods are available to identify associated variants?
So we didn't use our gene panels, but we did run a set of exomes looking at not BMI, but another complex trait, which was retinopathy, and it's sort of based on extreme cases. So there are methods that are available for doing association studies based upon NGS data. But these don't relate to our panels. They can and have been successfully applied with whole exome sequencing on modest numbers of cases and controls where you're looking at extreme phenotypes.
Yeah, I agree with Brian's comment on that, and to say that basically this type of work is being done by groups like Eric Schatz, but it's more toward a big data network that you're generating, but you have to have the phenotypic information as well. So unless you have access to that as you push through, you could, for instance, screen for variants affiliated with obesity, like we have in our panel, but for you to associate it back and predict BMI and so on, you have to have the control cases and the information to go there.
Okay, we have time for one last question. What are the main criteria used to filter out variants in your Oncomine?
Yeah, so the main criteria for filtering out the variants, as I presented in those Venn diagrams, what landed in the Oncomine Venn was specifically variants with known association with therapies or, you know, essentially actionability. So that number of, I believe it was 70, around 70 on a given run. When you go through and then you do a knowledge database, so when you run Oncomine, Torrent also has an Oncomine knowledge database. You can define features that way, meaning using their database, or you can define them using your own database, and essentially you're just associating known variants with known reported affiliations with disease, and that's how you reduce down from, you know, thousands of variants or hundreds of variants on a sample to just 70, or in some cases it's more like 2. So again, you can do that using commercial packages or internally using your own. And another thing that is important is upfront, before you do that, you should be filtering off the known information for your sample. So you noted we worked at a 5% minor allele frequency. You may want to work at like a 20% minor allele frequency depending on coverage, performance, and quality of sample, basically.
I would like to once again thank Dr. Meyer and Dr. Sebra for their presentation. Do either of you have any final comments?
No, just thank you for for hosting the session, and thank you to all the participants from wherever you are in the world.
Yeah, thanks so much, everybody. It's actually been really great, and really appreciate the time and hope it was exciting.
I would also like to thank our sponsor, the Clinical Sequencing Division at Thermo Fisher Scientific, for making today's educational webcast possible. For more information on Ion Torrent Next Generation Sequencing products, please visit thermofisher.com/Ion Torrent. Today's webcast will be available for on-demand viewing through 6 months from live date. You will receive an email from LabRoots alerting you when this webcast is available for replay. We invite you to forward that announcement to your colleagues who may have missed today's event. See you next time. Goodbye.